Bismillahirrahmanirrahim. Assalamu alaikum, dear students. I hope you all are well and fine. Dear students, I welcome you once again in Pakistan International School TAIF's virtual learning system for the session 2020. And this is first year class for the subject biology. Dear students, in today's lecture, we will talk about some more topics regarding chapter number 5, that is acellular life. In that, we are studying viruses, and today we will talk about some more viruses, that is HIV, human immunodeficiency virus, and the disease related that is AIDS. Let's start. What is HIV? HIV is actually human immunodeficiency virus. Human can live because this virus can only live in humans, and immunodeficiency means it damages the immune system of human on which it infects them, and viruses, that means it is retrovirus. So this is called retrovirus because it is having RNA, and then RNA from RNA it just make complementary copy of RNA, then it make DNA of that, and then it will incorporate it into the human genome. That's why it is called a retrovirus. uh, you just see the structure of that once again, that this is a circular globular structure. It is having envelope is having glycoprotein 120, that is this head, and the tail of this glycoprotein is made up of GP41, means glycoprotein 41, this one. Then inside it is having viral genome that is made up of RNA, and it is having certain enzymes also. Most importantly, that is having reverse transcriptase enzyme, integrase and proteases as well. Integrase and proteases as well, and it is having nucleotide core protein number 24 that is made the core of this genome. So if we see that HIV agent, that is RNA virus, it replicates actively into the T4 lymphocytes of the immune system. It is having a great and unique ability to destroy T4 helper cells. These are also called CD cell, and that is called CD4 cells or T4 lymphocyte cells. So once a person gets infected of virus, it remains in the body for lifelong period. So at that time, the person is symptomless, but after several years or after months to 10 years, it does not highlight its symptoms. HIV causes AIDS. AIDS is called acquired immune deficiency syndrome. That is actually not a disease, that is the syndrome in which lots of signs and symptoms of different diseases can be highlighted. So acquired means not born with, means that is not inherited, but that is acquired and transmitted from person to person. The next one, that it is attacks on body's defense system, like a human's body defense system or immune system gets weaker and weaker, as well as not working properly, that human immune system is not working properly, and that is a group of signs and symptoms that is called a syndrome. So AIDS may experience a wide range of different diseases and OICs. We will talk about them after some time. So what is AIDS actually? It is a disease in which there is a severe loss of body's cellular immune system and lowers in the resistance to infection. So what is the mode of transmission of that virus? That transmitted from blood products, vaginal fluids, semen, and breast milk. It means from the body fluids, it gets transmitted from one person to another. So that's why it is called STD, like sexually transmitted disease. How does HIV cause AIDS? Like uh, there is viral replication for 8 to 10 years, and lots of CD4 cells die in this process, and immune system gets impaired. So increased likelihood of opportunistic infections, that is called AIDS. Actually, AIDS is not the name of a specific disease. It is the combination of lots of disease. So in AIDS, uh, if we just talk about the normal person, so CD4 cells, their count is from almost 500 to 1500. 1500 CD4 cells per ml of the blood. But in HIV patients, it drops down less than 200 T cells or T lymphocytes or CD4 cells. So that is a very drastic condition for the person who is having this type of virus. So now let's talk about the HIV life cycle. HIV, this is the virus, HIV, that is having on that GP proteins, that proteins is 120 and 41. And here we are having CD4 cells in which these receptors are present, which are bound with these proteins, and this cell HIV that gets incorporated into the T cell. So life cycle of HIV depends on several processes. The first one is entry, in which there is attachment of the virus HIV in that, then binding with the T cells, and then fusion into that. The gene is incorporated into the host cell, and then reverse transcription occurs when HIV enzyme is used. That enzyme is called reverse transcriptase, that is utilized for the reverse transcription or of RNA into DNA. Then next step is integration, in which that HIV enzyme integrates, performs its function, and it joins or incorporate DNA of HIV into the host cell DNA. Then transcription occurs, in which HIV enzyme called protease performs its function, and it just uh, like makes the protein fragments in the cells, and then different fragments of HIV is being formed in T cells. Then there is maturation, when there is assembly of HIV parts, and then budding out of the host cell, and then maturation of the cell gets occur. So now we will see it through the diagram. Number one is there is free virus, which is going to attach with T cell. And then when it is attached, so attachment and entry is done. So virus binds to the CD4 cells, one type of core receptor, the CCR5 and CXCR4. That receptor is present on these cells. Look, this one, CCR5 and CXCR4 core receptor, which binds with these glycoproteins 120, and they are common on the cell surface of these T4 cells, and then virus fuses with the host cell. Number third is penetration. Virus empties its contents into the cell, like the genome and the proteins. These proteins disintegrates later on, but this envelope gets fused with the cell membrane of T4 cell. So this DNA gets incorporated into the T cell. Number 4 is reverse transcription occurs. Reverse transcriptase enzyme of HIV that works here and makes a mirror image of viral RNA strands to create double-stranded DNA. Then it creates the double reverse transcription, like first RNA is there, then it makes another RNA of like the copy of the previous RNA, then the like complementary copy of another polynucleotide chain is occur, and that then it forms DNA of this virus. Then integration, viral DNA is inserted into the cell's own DNA by integrase enzymes. Now this DNA is incorporated into the nucleus through nucleopore, and then it gets incorporated into the DNA of host cell by integrase enzyme. As you can see, HIV DNA and human DNA, now they are all together. When transcription is done in the next step, so the infected cell divides the viral DNA and long chains of proteins are formed. Now, because this virus does not have any type of ribosomes and any type of polymerase enzyme, so it just utilizes the enzymes and enzymes and ribosomes of or protein factory, we can say, of this T cells, and make the chains of proteins and required proteins. Now by a protease enzyme, this protein chain is broken down into several fragments, and then these fragments are just assembled and sets of viral proteins chains come together. At number 8, when budding off, now these are like there is core proteins, there is like DNA, and its other proteins are there, enzymes are there, so they all come together and assembled here, and after that they bud off by exocytosis from this T cell. This outer envelope is developed from this cell membrane of the host cell. Then immature virus breaks free of the infected cell, and lots of viruses are just burned off from that, and then they are free to infect other cells. Now maturation, the protease enzymes finishes cutting HIV protein chains into individual proteins. Combination of all these proteins make the core protein here, and like this copies of a virus is going to be produced in T cells. Now they are ready to infect the other cells. So rapidly, very rapidly, that is going to infect other proteins, other T cells. Okay, by this, like if we see some of the symptoms of this HIV, so HIV is having different types of symptoms. First, that it is having, number 1, it is having asymptomatic carrier. A means, as you know, symptomatic, like the person is not highlighting its symptoms, but he is the carrier of that of virus. So like within some days or some months, fever, chills, aches, and pains, swollen nodes, itchy rashes can be felt by that person, but after that they are going to be reduced gradually. and disappeared, and no symptoms occurs up to 9 months or some years. So but the person is affected and he is having no symptoms, but he is suffered from HIV, but he is a carrier. Number two, that is the now symptoms are going to be appear, and that is called AIDS related complex. AIDS related complex, and this AIDS related complex, the person is having some severe type of symptoms like swollen nodes in the neck, armpit, and groin that persist for several months, and other symptoms like night sweats, persistent cough, flu, diarrhea, loss of memory, inability to think clearly, loss of judgment, and depression can be occurred. And number three, the person can have full blown AIDS. So that is not appeared very soon, but it takes several months to several years. AIDS, full blown AIDS, in which the person can get opportunistic diseases like because the person is not having an immune system which is stronger enough to uh, to save him from other diseases. So these diseases just hijack that person, and they are very hard, or even there is no chance to recover from their diseases, no treatment is there. So that is very, very dangerous for the person. So more specifically, we will see about opportunistic diseases. Opportunistic diseases actually are the infections uncommon except when they are given opportunity to infect person, like an altered microbiota. Or we can also say that on the weakened immune system, the person is having more opportunity to get more severe types of diseases. So that can be life-threatening if a CD count is below 200 cells per mm cube, and that can lead to death as well. So opportunistic infections can be capsomyosarcoma, like you can see swollen tongue and degenerated tongue as well, and candidiasis, capsomyosarcoma, that is lesions and cancers of skin, and candidiasis of mouth, like swollen and ruptured tongue also. Not only these type of diseases, but women can also have bacterial pneumonia, herpes simplex, that is also the viral diseases, bacterial pneumonia, bacterial disease, and cancers of like reproductive system. There can be abortion, infertility, etc. What can be the treatment of AIDS? We can give now antiretroviral therapy. That is called ART, antiretroviral therapy. And now that is recommended for everyone, regardless CD4, T cells counts. Whatever the counts is, if like there is a specific HIV infection test, so if that is positive, so the person can get this therapy, but that is somehow costly and toxicities are there. So the person is required to strict daily pill taking, and can lessen quality of life. So there is the limitation. So the person scientists are just discovering some other types of methods to cure this one, but that cannot be cured properly. So when to start a treatment? When there are severe symptoms, opportunistic infections can be there. If count are less than 350 of CD4 cells, and there can be HIV related kidney diseases or hepatitis B or C in the presence of that. If the diseases symptoms, other symptoms, they can also suffering from that. So in antiretroviral agents, there are entry inhibitors like the drugs which inhibit the viruses to enter into the cells. Number two, reverse transcriptase inhibitors. The virus at the second stage, they inhibit the viruses to progress, like reverse transcriptase cannot bind and cannot produce reverse transcription of RNA. Protease inhibitors, like those drugs um, which are not allowing or stopping the protease to perform their function. And then here integrase inhibitors, they are these drugs are given and they affect integrase enzymes and inhibit them to perform their functions. So transmission prevention is very easy, like safe sex behaviors, and the person can have like if they follow the Islamic rules, then sexually transmitted infections can be we can control them. Avoiding injecting drugs, and disposable needles, syringes are to be used. Safe blood, ensuring blood transfusion must be safe. Cross matching must be done so the person can be safe from this all HIV related diseases. So we'll talk about the second very important viral disease, that is hepatitis. Hepatitis is actually the broad term that means the inflammation of liver. So it is most commonly caused by viruses, but also be caused by drugs, chemicals, autoimmune disease, and metabolic abnormalities. But here we will talk about the viral hepatitis. So there can be the 5 categories of virus: A, B, C, D, and E. So let's talk about them one by one. So we can see here hepatitis A, B, C, D, and E. They all are viral hepatitis. So hepatitis A is very contagious, and liver infection that can be caused by hepatitis virus A, that is called HAV. So that virus is having ribonucleic acid, just RNA virus, and we can say that enterovirus family. Like this just works as RNA, not as DNA, like we have just seen before. It can cause acute hepatitis with jaundice, and can cause liver failure. It does not cause long-term infection, and incubation period of that like virus is 3 to 5 days. It can be transmitted, if we see its transmission, so through fecal oral route, like if we just eat and drink hygienically, so we can be safe from this hepatitis. So personal hygiene, poor sanitation, poor personal hygiene, and contaminated food and water, that can lead to hepatitis A. So prevention is, some symptoms are fatigue, fever, abdominal pain, because that is in the liver problem, nausea, jaundice, killing of more red blood cells and producing less one, itching, sharp pain in the right upper quadrant of abdomen, anorexia. All these are symptoms of hepatitis A. Then we can take the measures like hand washing, proper personal hygiene, and environmental sanitation, control and screening of food, like active immunization. Active immunization means vaccines is given for its prevention. Hepatitis B, that can cause acute and chronic, both of them, and may last for 6 months without any symptoms also. So infected person are able to pass this virus during this time to other persons. Chronic hepatitis B, persistent, that can be having for 6 months or more after the acute infection of HBV. So mode of transmission is prenatal and permucosal, like from discharging of the body, and prenatal parenteral and permucosal, that can be from the mother to the child, that can be transferred, and from the blood products, as well as from contaminated needles, syringes, blood products, homosexuality, tattoo formation, and piercing in the body, that can lead to hepatitis B. It can be affect on all stages, and that can lead to psoriasis and hepatocellular carcinoma and worldwide. So there can be some symptoms also, that there is dark urine due to more degradation of red blood cells, as well as nausea, vomiting, fatigue, jaundice. All are almost similar like hepatitis A, but that is more severe. So hepatitis B and C both are having the same measures, like washing hands, avoiding toothbrushes and razors. So active immunization can be done because there is vaccination, and um so that can be a needle stick contact of mucus material, that can be also can be we have to be safe from all these type of situations which we have just seen before, as well as um, a blood transfusion should be screened and must be avoided. And interferon is the antiviral, we can see the proteins that is effective against HBV. Hepatitis C, that is a virus that is having RNA and incubation period 14 to 180 days. It means it does not show its symptoms in this time. So it is transmitted through blood products. Prior to 1992, it is also transmitted through unprotected sex and contaminated unsterile needles also. So it is found in IV drugs users and renal dialysis patients also, because at a very large scale these are syringes and these type of products are used for the blood transfusion. It can result in both acute and chronic illness, and chronic HCV infection results in liver cirrhosis, damage of liver, and the person is like suffering from a severe condition of liver cirrhosis, and there is no vaccine for HCV. So there can be the treatment that alpha interferon and ribavirin proteins, they are utilized in combination as a choice for chronic hepatitis, and there is no vaccine is available. So blood transfusion must be screened as preventive measure. Hepatitis D or delta hepatitis, that is a defective signal and RNA virus. This is, and it requires hepatitis B virus to replicate. Incubation period is 2 to 26 weeks. And chronic carriers HBV always risk of transmission. Source of infection are same as HBV, so they are mostly related with one another. So there can be the treatment and prevention and symptoms are as HBV. Now, hepatitis E, that is caused by HEV virus, and that is also RNA virus, and incubation period is 15 to 64 days. That is also transmitted through fecal oral transmission route is there. Source of infection is mostly contaminated water, poor hygiene, poor sanitation, mostly in developing countries like Asia, some of Africa, and Mexico as well. So that is more common in adults, and it can be severe for pregnant women. So hepatitis E usually resolves on its own within 4 to 6 weeks, and treatment focuses on supportive care, rehydration, and rest, so that can be transmitted by contaminated seafood, water, and there is no chronic condition almost, and there is no antiviral treatment and vaccine, but we can say its transmission can be stopped or can be reduced by its preventive measures by good hygiene process. Dear students, that's it for today. I hope you understood it, and inshallah, we will see you in the next lecture. Wish you all the best. Allah Hafiz.